science friends. Let's just take a moment to review. So what are the main pigments in the chloroplast? And so the main pigments are chlorophyll, A and B. Chlorophyll is really the main pigment, but we'll go ahead and delineate into A and B. And then also carotenoids is another important pigment. And even in addition to those, there are several more additional accessory pigments. Okay. Describe the overall steps of how the electrons flow in the light reactions. I'm going to come back to this in just a moment because I really want to draw a big diagram for it. But we'll say photosystem 2, electron transport chain, photosystem 1, and then another electron transport chain, and then Calvin cycle. That's kind of the basic flow, how things are moving around. And once they are in this intermediate step, they're being picked up by NADPH to take them to the Calvin cycle. So that's kind of a very basic steps. But again, later in just after we finish this lesson, I'm going to go through a large diagram to summarize photosynthesis because I feel like those were helpful in cellular respiration. So I want to talk about one alternative pathway. Some bacteria use this. This is not super common, but I just want you to be familiar with it. So here we have that electron transport chain 1 and the electron transport chain 2. Again, they're not really called 1 and 2. I'm just calling them 1 and 2. But we have the photosystem 2 and the photosystem 1. So this is in the light reaction. But some electrons do not complete electron transport chain 2. You see that it's grayed out here. After they use photosystem 1 and the electron transport chain 1, but they don't use photosystem 2. And so they just use this cycle here between the electron transport chains and the photosystems. And that's how they generate their ATP. This is thought to be the earliest form of cellular respiration because it's a little simpler and it skips some of these other phases that are not really responsible for the generation of ATP. Remember, ATP is generated in electron transport chain 1, but not in that second chain. Okay, so this is just an alternative pathway that some organisms use, specifically some bacteria. Not very many plants are going to use this. But we've talked about how plants can be photosynthetic, but some bacteria are also photosynthetic. And so some will use this course. So we know that the electron transport chain happens in the mitochondria in cellular respiration. We talked about that a lot. And then most recently we talked about it happening in the chloroplast during photosynthesis. So let's talk about what are the major differences in these electron transport chains. Are they exactly the same? How are they different? And this seems to me like it would be a really good free response type of question. I'm not saying it is, but just as a teacher that, because they're important, but they're somewhat different. So first of all, the type of phosphorylation. So in the electron transport chain, remember this is photophosphorylation. And so the electrons are coming from when the sun has excited those electrons within the chloroplast, and it's called photophosphorylation. In the mitochondria, we have the electron transport chain is oxidative phosphorylation. Sorry, my voice is weird today. Oxidative phosphorylation. Now, we also have substrate level phosphorylation, but that's going to happen in glycolysis and in the Krebs cycle. We're talking about in the electron transport chain, and so for the mitochondria and cellular respiration, the electron transport chain is the one that does oxidative phosphorylation. Okay, so where do the electrons come from? Well, in the electrons are being, maybe the best question is what is the carrier here? So in cellular respiration, the electron carrier is NADH. In photosynthesis, the electron carrier is NADPH. And that's the electrons that when these electron carriers are broken down, those are the ones that are going to be fed through this co-transport, this chemiosmosis. Okay. Where does the energy come from? So in photosynthesis, the energy, remember, is coming from the sun. In cellular respiration, the energy is coming from food or glucose, the sugars in your food. And so that's the source that we're really breaking this energy down or converting it to something we can use. Okay. In cellular respiration, we are breaking it down because the energy is a big molecule, a big glucose molecule, and we want to break it down into smaller sources. We've talked about that several different times. In photosynthesis, we're not breaking it down. We don't break sunlight down, but we're harnessing it, okay? We are converting it into something that we can use. We're absorbing it. So remember, cellular respiration is defined as an exergonic process because it releases energy. Photosynthesis is described as an endergonic process because it's absorbing that energy from the sun. It's kind of like charging your batteries, right? If you have like solar panels, charging a battery in your car, we're trying to absorb that energy from a secondary source and hold on to it so the plant has something to use for its energy. Because remember, the plant's not consuming sugars, but it has to be able to make sugars to support itself structurally, okay? And so where does it get the energy to be able to synthesize those sugars? Well, that's coming from the sunlight, but we have to be able to store it into a smaller energy, that ATP that the plant can use, okay? And then last, the direction or location of the hydrogen pumping. So remember, overall, the process in cellular respiration is happening in the mitochondria. In the plant, it's happening in the chloroplast. So in the mitochondria, in cellular respiration, we're pumping from the matrix into the intermembrane space, but then ultimately that hydrogen, the pumping is actually happening from the matrix into the intermembrane space. In the chloroplast, we are in the stroma and pumping out to the thylakoid space. And you can kind of see these diagrams drawn here as to where that's taking place. Okay, so those are the major differences in the electron transport chains. Other than that, they function very, very similarly. Okay, we're moving these electrons down the electron transport chain. Those electrons generate energy that allow for this active transport. Remember, that stage is the active transport because we're pumping from low concentration to high concentration, but then we build up that concentration gradient, and it's going to force the electrons back down through ATP synthase, just like they did in respiration, to be able to generate that ATP. Okay? And so the overall process is the same. We call it different. We call it photophosphorylation. Where it happens is different. But the process, the steps are the same. We have those electron carrier molecules in there, those cytochromes that are carrying these electrons to use them to generate energy to make ATP. Okay? So let's look here at the diagram of the light reaction, just to review a little bit from yesterday. So remember, the light from the sun is going to strike these pigment molecules right here. And that excites those pigment molecules. And it's like a hot potato, I told you. And so they pass those electrons between pigment molecules. So the energy is being passed between the pigment molecules. And ultimately, it's going to like the chief pigment molecule, which is called P680. That's this pair right here. And then P680 passes those electrons up here to this guy, which is known as the primary electron acceptor. Okay? Now, the primary electron acceptor is going to pass those electrons down this first electron transport chain. Okay? So we did photosystem 2. Now we're doing the first electron transport chain. going through the cytochrome complexes that make up those electron transport chains, just as they do in cellular respiration. Okay, so in the process, as those electrons move down the electron transport chain, we are going to be generating ATP. So the energy is going to pump the hydrogen ions out of the cell, okay? And then these hydrogen ions there that have been pumped out of the cell are going to be pumped back through the cell, through ATP synthase, through energy coupling, through chemiosmosis. Remember, it's an active transport going from low concentration to high concentration. And so that requires the energy from the electrons as they move down this first electron transport chain. All right. And then when they come back through to ATP synthase, they will generate ATP. Now, this ATP is going to be fed into the Calvin cycle. The light reaction is the charger of the battery. So if you think about like maybe a battery or a solar panel or something like that. During the day, you use that energy. Let's use a solar panel as an example. On a nice sunny day in the daytime, you are charging your batteries. You're charging your generator or whatever with the sunlight energy. But then at night, when the sun is gone, then you use that energy from your charged battery to provide electricity and whatever for your house at nighttime. And so that's kind of what we're doing here with the light reaction. And then the Calvin cycle, sometimes referred to as the dark reaction. We can't harvest energy during that dark reaction. And so we still have to have something to make it go. Now remember, the Calvin cycle is also where the sugar is released. And so to be able to build those sugars, we have to have something to build them out of, and some energy to be able to build, right? So cellular respiration overall is an exergonic process. We've taken glucose and broken it down into something, and it's released energy. But now photosynthesis is not that way. Photosynthesis is an endergonic process. We have to take energy from the sun and hold on to it. Try to use it to make something. Try to put it in, harness it in this battery, right, in the form of ATP. And so this is just the steps in the process to be able to charge those batteries to store that sun energy as ATP so that the plant has something to use to be able to make its sugars, okay? So ATP again is only generated in this first electron transport chain. So after these molecules move down the electron transport chain, then they go to this second special pair, higher up in the command chain, is P700 is the primary electron acceptor, okay? And so it accepts those electrons. Now, before it had accepted photosystem 2 electrons, it was also already going through this process, right? This right here. These electrons had been excited. It had played hot potato with them, passed them to there, and it had offloaded them into the second electron transport chain. So it was simultaneously doing that process as well. But additionally, it's also accepting the electrons as they enter from the first electron transport chain, okay? And then as they move down this electron transport chain, then that's where the NADPH is going to be generated. Remember, that's our electron carrier, similar to NADH in cellular respiration, okay? And NADH is also going to the Calvin cycle. And that's another way that we've harnessed that energy to be able to convert it into sugars. So if you know nothing else of cellular respiration, if you could not tell me the first thing about cellular respiration, we're not even talking about cellular respiration. Photosynthesis. Please know this diagram, okay? It can answer, if you truly know this, you can answer about all of the questions on the test, okay? Because this just summarizes it so nicely. The water comes in the light reactions, okay? And it's the energy from the sunlight splits the water and releases oxygen. Where do the hydrogens go? The hydrogens get tucked away in NADPH, okay? And ATP, which is made in the light reaction. Remember, we're charging our batteries in the light reaction. We're building these things that we can use in the dark reaction of the Calvin cycle. And then the Calvin cycle is going to give us back empty batteries, right, in the form of NADP and ADP that have to be charged back up again, okay? The Calvin cycle is taking in the CO2. Remember, the gas exchange is happening in the stomata. Same thing down here. O2 is being released in the stomata. And fixing, the process is called carbon fixation. Fixing carbon into sugar, okay? Using it because sugar is structural for the plant. Really important for plant structure, okay? So that's a really pivotal diagram. Let me get back to where I was. All right, so now we're focusing on the Calvin cycle. So CO2 enters the Calvin cycle, as we just showed in that diagram. It came down from the top, and it exits as sugar. Now, in that diagram, if you go back and look at it, and I'm not going to, I wish I'd have pointed it out then, but it takes a long time to get back there. It just says sugar, CH2O, okay? So actually, the Calvin cycle does not truly make glucose. It makes a molecule called G3P, okay? Now, G3P is later processed in a separate process that's not technically part of photosynthesis to make glucose and other sugars. But the sugar that's technically made in the Calvin cycle is G3P, okay? Which is a 3-carbon molecule that can be put together to form that 6-carbon glucose, essentially. Okay, so in order to make one molecule of G3P, we have to have the Calvin cycle, the round and round, happen 3 times. 3 carbon dioxides, because it's a 1-carbon molecule, and we're trying to make a 3-carbon molecule. So it has to go round and round 3 times to be able to make 1 molecule of G3P. And in the process, in those 3, okay, so this is per 3 rotations, per 1 G3P, not GTP, sorry. It's going to expend 9 ATP and 6 NADPH. That should have a P in it. NADPH, okay, is what it's going to use. And remember, those came from the light reaction. They were generated in the light reaction, okay? So, in carbon fixation, we've got our CO2 that's coming in from the air, right? And it is combining with a molecule called ribulose bisphosphate. With an enzyme called Rubisco. Now, that is a tricky enzyme because it doesn't end in ACE like most of our enzymes do. All right, and so ribulose is a 5-carbon molecule, and it is attaching with this CO2, and so it makes a 6-carbon intermediate that's really highly unstable that actually only lives for like a second. You don't really need to know that. I don't really care so much that you know, really honestly, ribulose bisphosphate. But Rubisco is somewhat important. It is thought to be the most abundant protein on the planet. Is Rubisco that's involved in the photosynthesis chain, that's involved in this Calvin cycle. So in the Calvin cycle, we have a series of carbon fixation. We're trying to fix this carbon, fix it in with ribulose bisphosphate, using the enzyme Rubisco to form this intermediate, essentially here, okay? And so then this intermediate is reduced, okay, to become other molecules, all right? And I'm going to show you a diagram. It's going to make this a little more sense in just a second. And then ultimately, we're trying to get back to this ribulose bisphosphate so that we can accept more CO2 in. Okay, just the same way as we did in the Krebs cycle in cellular respiration. We had that oxaloacetate, we were bringing in acetyl-CoA, and we were in the process releasing ATP and NADH. Here we're releasing ATP and NADPH, and in respiration we also had FADH too. But we're trying to regenerate this guy so that we can accept more CO2, so that we can keep this process going, okay? All right, so here's what we've got. The Calvin cycle is, here's our ribulose bisphosphate that's going to bind or join with the CO2 using the enzyme Rubisco to produce this short-lived intermediate here, okay, which ultimately is converted into all of these different things to try to make ribulose bisphosphate again so that we can bring back in more CO2, okay, because the more CO2 that we can put in means the more of this output, and these outputs, well, actually those are not outputs, sorry, but this output that we can get, okay. These are not outputs, these are inputs. These are used in the process. These came from the light reactions, okay? All of these came from the light reactions. And they are used in the dark reactions. They're broken down. Their energy is harnessed to try to make this guy, because that's the whole purpose of the Calvin cycle, is to make this sugar. We got to make this sugar. This is what the plant needs. And so it takes energy to do it. That energy comes from the light, which we harvested in the form of ATP and NADPH in the first step, the light reaction cycle. And so now we're using that energy to make the G3P, okay? And the faster we can do this Calvin cycle, the more CO2 we can bring in, the more G3P that can be made. Okay?